Hello all, my name is Ryan Abdelaziz and I am presenting my initial discussion for the VoiceThread 4 assignment on ethical considerations in biotechnological experimental design. So, for the first question, what ethical tension biotechnology is not taking seriously enough? I would identify the expanding secondary use of donated human tissue and genomic data. So this is essentially relevant to my position in postmortem brain collections. A donor may consent to neuroscience research, but that tissue can later be used for whole genome sequencing, multi-omic studies, data linkage, or technologies that did not exist when consent was obtained. Because donor is deceased, it can be easy to assume that the ethical risk has ended. However, genomic data can reveal information about relatives, psychiatric or substance use findings, uh, which may in turn affect families, and research conclusions can stigmatize broader communities as a result. So the tension is not simply between privacy and scientific progress. Scarce human tissue should be used to generate as much valuable knowledge as possible. The deeper question is whether broad scientific use remains consistent with the donor's consent, values, and expectations over time. I think this issue is minimized because consent is often treated as a completed form rather than an ongoing process. Scientific incentives also reward large data sets, open sharing, and rapid reuse, while careful consent management, data governance, and community engagement require resources and receive less recognition. For the second question, do ethical constraints on research strengthen or weaken scientific rigor, or is that framing itself a category error? I think that framing is largely a category error. Ethical rigor and scientific rigor are usually complementary. So, for example, fair participant and donor selection directly affects external validity. If tissue banks and genomic data sets are overrepresent one ancestry group, resulting conclusions may not generalize an entire population. It's both an ethical concern and a scientific issue. Ethically, it's a problem because of unequal representation, and scientifically, it's an issue because of bias sampling. Consent and data governance can also improve reproducibility. Clear records of how tissue was collected, what clinical information was linked, and which users were authorized strengthen provenance and determine whether data sets can be combined responsibly. In post-mortem research, ethics also influences measurement validity. Psychiatric diagnosis may be reconstructed from medical records, toxicology, and interviews with relatives. Researchers should not present those diagnoses as more certain than the evidence allows, because an analysis requires a clean category. So preserving the uncertainty is both respectful to the donor and scientifically accurate. It does not mean, however, that every regulatory requirement improves science. Some processes become repetitive or overly administrative, but that is a distinction between meaningful ethical safeguards and procedural burden, not a conflict between ethics and rigor. So for the third prompt, where should the line fall between an individual researcher's ethical judgment and institutional or regulatory oversight? Who should hold final authority and under what conditions? Where should the line fall essentially between individual judgment and institutional oversight? So I believe researchers should hold primary responsibility for identifying ethical problems, but they should not always have the final authority. Or the individual researcher should not have the final authority. So researchers are closest to the methods and may be the first to recognize that a new analysis can create privacy risk or exceeds original consent or could harm a family or community. They must be willing to raise those concerns and pause the work when necessary. Approval should never replace personal ethical judgment, in my opinion. However, independent oversight should have final authority when there is ambiguous consent, significant re-identification risk, vulnerable populations, conflicts of interest, controversial use, or potential harm beyond just the individual donor. A researcher may be strongly invested in completing a study, so that person should not be the sole decision maker in these sorts of high-risk situations, which is why I say that the final authority should not just be the individual researcher, especially under conditions where it's a very high-risk sort of study, or where the researcher has spent quite a bit of time with the research and might be anxious to kind of get it out no matter what. So for post-mortem collections, I would support a layered model where researchers make routine decisions within clearly approved boundaries. Biobank committees and IRBs evaluate new or uncertain secondary uses. When research could affect identifiable communities, donor family, or community perspectives, should also be included under this broader authority. Overall, my position is that ethical oversight should establish enforceable boundaries while researchers remain responsible for ethical reasoning. Within those boundaries, the strongest biotechnology research is not research that advances despite ethical constraints, but research whose scientific and ethical designs can be defended together and are both intentional. So that is it for my initial post. Thank you all. I look forward to the discussions later on, and I hope everyone has a wonderful day.